So the next speaker is going to be the co-chair of this course and symposium. It's really great pleasure to introduce my conspirator in arms, in academia, in every way, a great, phenomenal colleague who's a leader in the field of cognitive neuroscience and neuroimaging, Susan Bookheimer. She's the Joachim Fuster Professor of Cognitive Neuroscience, Director of the Staglin Imaging MRI Laboratory. She's the Director of the Intellectual and Developmental Disabilities Research Center here, and the co-director of one of our big network grants in autism, our Center for Autism Research and Treatment grant, which is a NIH Center of Excellence. So it's with great pleasure that I introduce Susan Bookheimer, and now the computer will work.
Okay, thank you so much. I've been asked to speak today about clinical perspectives in brain imaging, and this turns out to be very difficult to cover all the cool things that have happened in the last year because there are so many things, many ways of looking at brain imaging, structural MRI, functional MRI, and that includes task-based functional MRI and also resting state, which I'll touch on today, as well as brain chemistry using a technique called MR spectroscopy. And among the clinical uses that we are thinking about and mostly doing research on still at this stage are understanding the causes, early detection, outcome prediction, as well as looking at the effects of intervention. So I really don't have time to talk to you about all of the cool things that have happened in the last year, but what I will do is highlight a few key studies that let us know where this field is going, what kind of things we're able to do, and to hopefully give you some ideas about how we're going to be able to use brain imaging technologies in the future to inform us about diagnosis of autism and to help us with treatment and measuring the efficacy of treatment. So the first thing I will talk about then is understanding the basic mechanisms, the causes of autism using MRI spectroscopy, which is a way of looking at brain chemistry. So as a background, one of the prominent models of autism that Dan already alluded to is this idea of excitation and inhibition imbalance in autism. This is something that has come from studies of genetic syndromes and studies of animal models, and it suggests generally that there is not enough inhibition and too much excitation in the brains of individuals with autism. So MR spectroscopy is an MRI technique that allows us to see a particular set of brain chemicals, not all the brain chemicals, but some of them, using MRI. And there are some brand new techniques that are out there that enable us to measure GABA, which is one of the brain's primary inhibitory neurotransmitters, as well as glutamate, an excitatory neurotransmitter. So this study, and the reference is at the bottom here of the slide, is a relatively small study of 15 children with autism and 13 typically developing adolescents using this NMR spectroscopy. So in the spectroscopy, you find one sort of big old square chunk of brain to study. So you only get to scan one little piece of the brain, and you have to keep scanning that same little piece for a long time in order to get these chemical signatures. So this particular group looked in the occipital lobe, and they chose it just because it gave the best spectral quality. The occipital lobe is a pretty homogeneous area of cortex. Other areas of the cortex might be smaller and have more white matter or more cerebral spinal fluid in it. This will give you a nice clean signal. And they used first this standard MRS technique, which is called PRESS, and it generates a glutamate peak. And there's this newer technique called megapress, which generates a GABA peak. So what they did in this study was to use both techniques on the same kids and look at both concentrations. And so one of the things they found was a higher glutamate concentration in the kids with high-functioning autism and also lower GABA concentration. So that suggests that there is this imbalance in excitation with increased excitation and decreased inhibition in the brains of children with autism. Very consistent with the animal models, and this really gives us a tool to now compare what we see in the brains of humans with what we see in the brains of animal models. As we develop treatments with animal models, we can then apply them to humans, and we have a way to measure that. And this is a study using this kind of approach, looking at the effect of pharmacointervention on this brain chemistry to see if an intervention can target that very mechanism that we're talking about, this excitation, inhibition, imbalance. So this is a study of young adults with autism, 17 with ASD and 20 controls. And the reference, again, is at the bottom. And it challenged the system with this drug called Riluzole, which has a broad range of actions at both the GABA and glutamate receptor levels. Specifically, Riluzole blocks the presynaptic release of glutamate and facilitates GABA-A receptor activity. So the ultimate effect of this drug should be to reduce excitation and increase inhibition. And the question is, will it have this effect in the brain of individuals with autism? So they used a different MRI technique. They only had press. They didn't have the cool megapress technique. And you can see they've also used a different region of the brain. And contrary to the prior study, they didn't find any baseline differences in the glutamate to GABA ratios in autism versus controls. But what they did do is they scanned kids one day without drug and another day with drug and compared the glutamate and the GABA levels. And this drug, Riluzole, increased the proportion of GABA in the prefrontal cortex of the ASD group, but decreased the proportion of GABA in the control group. So there's a group-by-drug interaction. Here's another way of looking at the same data. At the corresponding data. So they also did what we call a functional connectivity, in which you find a seed region. In this case, here it is. It's the same region where they put the MRI spectroscopy. And they asked the question, how does the rest of the brain functionally connect to this seed area of the brain? So if you scan using a functional MRI for individuals at rest for a period of a few minutes, in this case about 5 to 6 minutes, you look at regions of the brain that correlate over time, so their activity changes in sync with one another. Those areas of the brain are likely to be very connected. Other areas of the brain that are unconnected, one area might be doing one thing. It's really hard to do this, by the way, and the other area doing the other. It's like patting your head and rubbing your stomach at the same time. So that would be a set of regions that are uncorrelated in the brain. And so what they found was that there were significant differences between control children and children with autism in the connectivity between this medial frontal region where the spectroscopy seed was and these other areas of the brain that are shown here. So this is the significant difference between the groups. But then they compared each of the groups on drug and off drug, and here are the results. So this is an area of negative connectivity, and you can see that it's negative in the controls on placebo and also very similarly negative in the controls on this drug. But we can see that among the individuals with autism, at rest, there isn't this negative connectivity, and with the drug, it restored that connectivity to the same levels as controls. So what this suggests is that if you have a drug that's targeted at the basic mechanism underlying this aspect of autism, excitation and inhibition balance, that it changes brain function in a way that looks like it's normalizing the brain function. So this is very exciting. There are some caveats as well. Since there were no differences in baseline EI imbalance, we have to wonder why that is. And it could be because there's a small n, and it could be because they used a different region of the brain, and it could be because they used the less sophisticated MRS method. They used press, but not the newer megapress. But on the other hand, they did show that the balance of excitation and inhibition could be altered by a drug and could be altered in a way that normalized, and that individuals, and these are adults, could respond to this kind of medication that's targeted at the mechanism. So this is a very important advance in autism research. Predicting autism diagnosis is another area of great interest, and this particular study used resting state connectivity to do so. This is an interesting group. There is an ongoing network funded by the NIH called the IBIS, or Infant Brain Imaging Study Network. They've been collecting a large cohort of 6-month-old data in a multi-site study. So 4 sites around the country are collecting MRI data, starting at 6 months and then following these kids up to 1, 2, and 3 years of age. So they just came out with a new study this year looking at resting state functional connectivity, the same kind of connectivity that I just described to you, in infants of 6 months of age that were sleeping. And then they followed them until they were 24 months, did a diagnostic test, and looked to see what kids ended up with autism and compared those who did end up with autism to those who did not. So they had 15 infants at high risk for developing autism, and in the end, 11 of them ended up having autism. So this is the way it works. First, you start with a bunch of regions in the brain. So here is a wide number of regions in the brain. And from this whole set of regions, they chose those that had previously been implicated in autism. And they collected then fMRI data while the babies were sleeping for about 5.5 minutes, and then correlated the activity again over time in each of the regions. And what they do is generate a big correlation matrix for each subject, and then correlated that with outcome at 24 months, whether they got the diagnosis. And they found those subset of regions whose connectivity, the correlations between these different regions, related to later diagnosis. And so when you do that, you do a principal components analysis, and you pull out that whole series of connections to generate these principal components. And this is the first principal component, which did a very good job of separating the groups. So this is the performance on that principal component. So it's a whole range of brain correlations. And all of the typically developing kids ended up over here. They were correctly classified. And 9 of the 11 kids with autism were correctly classified, and there were 2 somewhere in the middle. And then you can look at then what are those connections that made those prediction, and relate them to some behavioral measures. And here I have just shown a subset of the data showing the relation to the Mullen scale as well as the repetitive behavior scale. And so the blue and red refer to positive connectivity and negative connectivity. The width of the bar refers to the strength of that connectivity. And so what we're showing here then is that there are just a certain subset of regions that are more positively connected in autism, and others that are more negatively connected that predict the outcome, and particularly how it relates to specific behaviors. And there aren't any really strong patterns here. There are some cross hemispheric connectivity differences and some top-down connectivity differences. And this kind of research has some problems associated with it. Specifically, it's a small N study, first of all, and only 11 kids developed ASD, so it's hard to say how reliable these results will be. The classifier itself is not cross-validated with a second and independent sample. So you can always classify based on the data that you have, but the real question is, if you apply the same classifier to a new set of data, will you get the same results? And they haven't discovered that yet. That's something that we hope will be done in future research. Then the relation to ASD scores is positive, but of course, in a sense, that's by definition because the scores on those tests are part of the autism diagnosis. But nevertheless, in general, these kind of data suggest that abnormal brain connectivity at 6 months of age could potentially predict autism diagnosis 2 years later. And that's very important for us when we think about early intervention, right? We want to be able to intervene as early as possible. And if we can find some brain markers of autism earlier in life, that will make it that much earlier than we can think about interventions. Of course, 6 months is far along in the process. As Dan mentioned, we believe that the abnormal connectivity and abnormal development of the autism brain begins probably before birth and in the first few months of life. And then a little bit, Dr. Morella DeProto will talk to you about work with infants who are only 6 weeks of age. Now, clinical subtyping is something that's also very important in autism. Dan has talked to you about the value of precision medicine. And from a human imaging perspective, what we'd like to be able to do is to identify subtypes based on the brain. And so there have been some new attempts to do this using structural MRI. The history of structural MRI in autism is difficult and fraught with varying results. There have been many imaging studies. Some have found things like the cerebellum was too small, or overall the brain is too enlarged, or only some structures are enlarged, or the size or the amount of enlargement of structures depends on age. It's larger at birth and smaller later on in life. There's a lot of inconsistency across the literature. And one of the questions is, could this inconsistency simply be because there's a lot of heterogeneity in autism? And there are different kinds of autism. And we know from the genetics that Dan talked about that there are many causes of autism, and they should have different brain mechanisms. So Dan also mentioned big data, and this is also very important in human neuroimaging. And one of these big data initiatives is called the ABIDE Data Initiative, the Autism Brain Imaging Data Exchange. They've collected structural MRI data from across the world, including from our group. And this particular study drew from that large data set to find 220 children, 150 males with autism, and the rest typically developing males, and all of them are tested with ADOS and ADI. And they did a very detailed analysis of the structure of the cortex using 4 different metrics, and I'm going to talk about what those are. So there are many ways to look at brain structure. One is to look at cortical thickness, which is a measure of the density of the synapses. One can look at intensity blurring. That means how sharp is the boundary between white matter and gray matter. And if it's not very sharp, it suggests that the neurons may not have migrated as smoothly as they're supposed to be. Cortical surface area, which has to do with how much gyrification there is, may be sensitive to alterations in the columnar organization. And I'll show you a picture to make this a little bit more clear. And then a fourth measure, which is a brand new one, is called geodesic distance, which is the shortest path between different points on the cortex. And it's supposed to represent cortico-cortical connectivity. And that makes a little bit more sense when you see it in picture form. So cortical thickness would be this measure, just what is the distance between here and here in every part of the brain. And that's sensitive to this laminar organization. These are all the layers of the cortex. This is the white matter, this is the gray matter. There should be a nice sharp boundary, so intensity blurring has to do with how sharp the boundary is right here. Cortical surface area is all of the surface area of all the gyri and sulci. And then geodesic distance would be the distance between here and here, which is reflected in these cortico connections. So thickness would be the cortical to subcortical connections, and the geodesic distance would be the cortico-cortico connections. So what they did was they got all these 4 measures, and they did a procedure called a clustering analysis, where they take all these 4 features at every single point in the brain and create a massive matrix for each subject. So each subject will have a profile across each of these 4 metrics, and then they use this hierarchical clustering technique, which is a way of grouping subjects according to their structural profiles. So this is sort of roughly what it looks like. I know this is a little hard to see, but I'll show you a close-up in a second. So this would be the brain picture. They would take a little patch. They put it all in this connectivity matrix. You can see these are the 4 different techniques that we talked about, the thickness, the intensity, contrast, and so forth. And they clustered the data to find groups that are similar. And they came up with 3 different brain profiles of autism, which we'll just call autism 1, 2, and 3. And so these profiles show differences on different measures. So for example, in ASD1, cortical thickness is greatly increased, while it's decreased in ASD2, and it's not changed in ASD3. In contrast, intensity is reduced in ASD1, increased in ASD2, and doesn't change in ASD3. I'm just going to show you one close-up just to get an idea of what that looks like, so you can see how different the profile of cortical thickness is between just these 2 groups of ASD kids, and how different that is in turn between the intensity contrast. So looking more broadly across these 3 subtypes, we have one subtype where there is diffusely increased thickness. This is sort of the big brain cases, where they have some cortical interface blurring throughout the cortex, really, but no functional connectivity changes, whereas subtype 2 has widespread thinning in the cortex, particularly in the parietal or back of the brain. They have sharp edges and decreased distances, and marked connectivity decreases. And subtype 3 has only selective increase in the distance of certain brain areas involved in the insula, which is a deep limbic area of the brain, but marked connectivity differences. And these different subtypes also have some differences in their profiles. For example, here's the ADOS profile, and you can see that ASD1 is a less severely affected group. But I think what is here's a better picture of this. You can see up close. These are the different ADOS scores. These are the different sub-measures of the ADOS, and so social interaction is less impaired in group 1 than it is in group 2 and 3, but repetitive behaviors are different in group 2 compared to groups 1 and 3. So this is very interesting because it is basically a first step to bring us from brain structure into subtyping. Now I'm going to move on to functional MRI and the effects of intervention. So what we'd really like to do is to use our brain data to establish the efficacy of different kinds of interventions. And this study did, this study looked at intranasal oxytocin on reward circuitry in autism. Now, Dan talked to you a little bit about oxytocin as being one of these important pro-social hormones. And a lot of parents now are giving oxytocin nasally just through a little nasal spray. It has a very short half-life. It only lasts about 20 minutes, so these kids are spraying all day long. With the belief that it will increase social rewards, increase sociability, increase social bonding, and there's good animal evidence to suggest that this is the case. There is some evidence in humans using functional imaging that oxytocin increases things like the sense of trust. And Dr. DePretto's group has done some extensive work on the oxytocin receptors in humans and how that relates to autism as well. What this study did was looked at the effect of treatment on brain circuitry, and specifically they were looking at reward circuitry. So one of the models of autism is that they do not seem to respond as well to rewards. The reward system is just not as reactive as typically developing individuals. And if the reward circuitry isn't driving your behavior, then there isn't so much motivation to go out into the social world. So we call that the basis of the social motivation hypothesis of autism. So in this group, they've looked at 28 children and adolescents with autism, and they did an fMRI task called an incentive delay task. So what they do is they scan their brains, and while they're scanning their brains, the children are playing these games. They see a symbol, and some of the symbols suggest that if they respond really, really fast, they'll get a reward. And if they don't respond fast enough, they won't. Other symbols, they don't get a reward, so they don't care as much about it. And you can compare then the brain responses to those symbols that they know are supposed to give them a reward to those that are not going to give them a reward. And that's a measure of how much the brain is responding to reward. They use two reward types, non-social rewards, which are money, and social rewards, which is just a smiling face. And they scanned a couple days apart to make sure, with and without oxytocin, to make sure that there wasn't any oxytocin carryover effects. So what they found was, comparing oxytocin versus placebo, they saw increased activation in the reward network. Here is where it is. So this is the nucleus accumbens area, and then two other areas, medial prefrontal cortex and lateral orbitofrontal cortex, and all these are involved in the system that does sort of the inside reward sensations with the brain's processing of that rewards. Unfortunately, they found these significant effects for the non-social rewards, for the money rewards only, and not for the social rewards. So it wasn't consistent with the hypothesis that it would really increase sociability, but it was consistent with the hypothesis that it increased reward responsiveness. And so it's possible then that we could imagine that this intranasal oxytocin might make the kids more reward responsive, and that might be important in a behavior modification or a learning context. So just to wrap up, this is just a very small sample of the new imaging research that has happened in the last year, but there's some major points. First, we are benefiting from big data initiatives, these data sharing efforts. This is true in imaging as well as in genetics. Also, the data have suggested that brain imaging can identify basic mechanisms that are consistent with animal models. These brain imaging techniques can allow us to test the effects of treatment on the brain rather than just on behavior, and the brain may be a more sensitive measure of the effects of a drug than behavior is. we can use brain imaging perhaps to identify risk for autism early in life and maybe even to predict future diagnosis in babies. So ultimately, this should facilitate early interventions and evaluation also of the efficacy of interventions. So with that, I will thank you very much.